Okay, so this is the lecture we're going to go through some DNA replication. Right. So here's my presentation here, and let's make this full screen. Okay, so here's a really good picture. I mean, looking at this, it has a lot of the different things that that we need to be able to explain when we go through DNA replication. And it shows the difficulty of the lagging strand in comparison to the leading strand. Okay, the leading strand is just going the same way helicase is. So as this unwinds, it's just going to keep following behind it. This is the problem. Okay, this is the problem right here, is the lagging strand. So the first thing, we got to make sure that we understand the 5' to 3', because this is going to be very, very crucial when we start talking about DNA replication and understanding the difference between the leading and the lagging strand is this identification of what 3' and 5' means. So DNA is a double helix, okay, and it's said to run anti-parallel. It means one strand is running in one direction. And really, if you look at the sugars, that's the easiest way. We got upright sugars on one side, and we got upside down sugars on the other side. Okay. Now, we've already gone through this when I talked about the structure. So if I'm going a little bit quick through the 5' to 3', you may want to look back at the DNA structure lecture I gave to identify what 3' and 5' is. Okay. And then some other things I would expect you to know is that between guanine and cytosine, so between G and C, I'm going to have 3 hydrogen bonds. And then between A and T, I'm only going to have 2 hydrogen bonds here. Okay, so these are some videos that I would suggest watching. Okay, it recaps on compactation. They're just really, really good videos, and especially when we get into the replication and protein synthesis, the videos are crucial to understanding. It's very difficult to just explain in words exactly what's happening. Okay. So let's get into DNA replication. Now, there's 3 main phases in DNA replication, and I'm going to kind of focus on those 3 phases. We've got the initiation phase, preparing DNA to duplicate. Okay. Then we got the elongation phase, which is actually adding the base pairs to the 2 templates. And then finally terminating it, ending it. The 2 new strains of the DNA are complete and are separated. So we're going to go through each of these phases one by one, starting with the initiation phase. So here we go, initiation phase. Okay, so replication cannot begin anywhere on the double helix, but only in certain spots called replication origins. So if we look at, say, here's our DNA, so here's one strand and here's our second strand. Okay, here's several different replication origins. And what I mean by that is that you can't just start replicating DNA. DNA is not going to unwind. And here we have kind of a picture of it unwinding. It's not just going to unwind anywhere that you want. There's certain spots that it's capable of unwinding. And those spots that it's capable of starting to unwind are called origins of replication. And notice that we have a ton of them. Okay? We don't start at one spot and then replicate our DNA from the beginning all the way to the end. What we do is we start replicating it in several spots all at the same time. So I'm going to start replicating it here, here, and here. And you can see that we have a whole bunch of replication bubbles. So when we have to replicate our DNA, it would take way too much time to just start at one replication origin and then go all the way through replicating our DNA all at one time. Instead, we replicate it at several different spots all at the same time so we can duplicate our DNA much quicker. Now, the replication origins are identified by a certain base sequence. So within this sequence, you're going to have a certain series of letters. These are not the series of letters, but this is an example. We have a certain series of letters, and that tells the DNA that this is a replication origin. And then I'd have that same series of letters, say here, this series of letters here, this series of letters here, and that would tell the DNA on where to start replicating. Now, the replication origin is where DNA is going to begin unwinding, and as it unwinds, it makes a replication bubble, and that's what we call here. Okay, they're just kind of little bubbles within the DNA where it's being replicated. And then at the very end, we have something like this. We've got 2 new pieces of DNA. Okay, so the blue was the original piece, and then the red is the new piece, and now we have 2 new pieces of DNA. Okay, now once the replication origin has been found, 3 important proteins get involved. We're still in it during initiation, but now we're going to get 3 very important proteins. Okay, first thing, okay, we're going to have helicase, which breaks down the hydrogen bonds between base pairs, the AT bonds, and separates the 2 strands. It usually starts with an AT. Okay, an AT is usually a little bit easier to break because you've got the 2 hydrogen bonds. It doesn't always, but it tends to start with the AT. Okay, but we're going to start now ripping apart all of these hydrogen bonds. So here's my helicase, and I'm just going to go through and start breaking all of these bonds in here, okay, where the bases are bonded. The second thing is DNA gyrase, also called topoisomerase 2. They're both the exact same things, just 2 different names for them. And the whole point of this thing is just to relieve the tension caused by helicase. If I'm starting to rip open the strain down here, up in this area right here, it's going to have the tendency to start ripping open before helicase gets there. Just because you have this strand going this way, this one going this way, so there's a lot of tension right in front of helicase. And it has a tendency to start ripping the DNA before helicase gets there. So we have another molecule that kind of just relieves the tension, okay, and that's called DNA gyrase or topoisomerase. Okay, and it's just relieving the tension in front of helicase. And then the last thing we have during the initiation is the single-stranded binding proteins, and they're just going to kind of line the already open DNA to prevent it from going back together. Okay, if we didn't have these, as soon as helicase kept down to here, this strand and this strand would go right back to each and bond with each other. Well, we don't want that happening because we haven't replicated DNA yet. So single binding proteins really just kind of keep the DNA from binding back together. And those are the three main proteins that get involved during initiation. Okay. Now, when we talk about replicating DNA, we're only going to start, and we're only going to talk about it in one direction. But when you have a replication origin, it really goes both ways. I'm going to start duplicating DNA this way, and I'm going to start duplicating DNA this way. Okay. Now, this gets way too confusing to be able to talk about everything at the same time. So the whole time we talk about replication, we're going to be talking about half of it. So if you look at this black line, everything to the right of the black line, we're going to talk about just kind of one side of the replication bubble. It would get too confusing to talk about both sides of the replication bubble at the same time. So everything we talk about replication, it's only going to be on one side here. Okay. So here's kind of a great picture of everything that we need to be able to talk about. So here's our DNA gyrase or topoisomerase, okay? And this is during the initiation phase. We got single-stranded binding proteins during initiation and helicase all during initiation. So here's our three molecules, okay? We're not dealing with any of this stuff yet during initiation, but we're only dealing with this molecule relieving the tension, helicase breaking it apart, and then single-stranded binding proteins preventing it from going back together. And that's it for initiation. So now let's go through the elongation phase when we actually start adding bases. So during this phase, new nucleotides, bases and sugars and phosphates are added to each parental template strand. So the original strand, if we go back here, so here's my original strand, what we call the template. Okay, it's also called the parental strand. And then this is my new strand that I'm creating. Okay. So the new nucleotides are required to form the brand new DNA strand came from recycled dead cells and from the foods we eat. When we eat a banana, we take the banana's DNA, we take all their A's and their T's and their G's and their C's, and we take their phosphates and their deoxyribose sugars, and we start attaching it and making it into our DNA. So we simply take the bases, like when you look at, say, your own DNA, and if we go back to, say, one of these bases, if that's an A, in your DNA, that A used to be inside a plant, or it used to be inside an animal. We basically just keep recycling these bases from the foods that we eat. Okay. Now, this is probably one of the most important parts right here. Okay, the nucleotides can only be added in a 5' to 3' direction. So I can only add going from 5' to 3'. I can't go the reverse direction. Okay, and that's a very, very important point. It's what leads to the problems in the lagging strand. Now, the fact that nucleotides can only be added in the 5' to 3' direction creates one strand of DNA that it's easy to duplicate, which is our leading strand, and then one strand that is much more difficult, the lagging strand. So we're going to start with the leading strand. Here's kind of a picture of what's happening here. So if we look right down here is our leading strand. It's going to be very easy to duplicate. Helicase is just going to keep breaking open, and DNA polymerase is going to keep adding bases, and we just keep adding bases. So we're going, to the left here, and this is going to keep opening up, and I'm just going to keep making it. Very, very easy. Okay, a very easy process. Because, and this is the big thing, if you look at the parental strand, and the parental strand is kind of in green here, the parental strand is 5' to 3'. Okay, now across from my 3' is I'm going to have a 5' of my new strand. And because DNA polymerase works in the 5' to 3' direction, I can just keep adding bases here. Now when I go up to the lagging strand, if we look, I've got my lagging strand is 3' following all the way up into 5'. Okay? So that means that DNA polymerase is going to have to work in this direction, the direction that I'm moving my mouse, so to the right. So it's going to have to keep adding bases this way. Well, the problem is that DNA helicase is going that way. So DNA polymerase on the lagging strand right here is going the opposite direction. It's going to start heading to the right, and helicase is heading to the left. So what's going to have to keep happening is DNA polymerase is going to have to break off, come back, and then start adding bases here. And then helicase is going to move. It's going to have to break off, come, and then keep adding these little segments. Okay. So let's start with the easy strand, the leading strand. Okay. So here we got the parental strand is in blue, dark blue. Okay. And if we look on the parental strand, I have it oriented in the 5' to 3' direction. Okay. In helicase, if you look at it, helicase would be right here, and it's heading towards, on the leading strand, it's heading towards 5' on the leading strand and 3' on the lagging strand. Now, I know these numbers are confusing, but we have to get these down. Okay, so if you kind of remember that helicase on the leading strand will head towards 5' and it will head towards 3' on the lagging strand. Okay, so because I have my parental strand in the form of in the leading strand, because I have my parental strand in the 5' to 3' direction, my new piece of DNA can now start being added in the 5' to 3' direction, the direction that we want DNA polymerase to go. Okay. So the parental strand is in the 3' to 5' direction. If you start here, here's 3' to 5'. The newly synthesized DNA strand will be done in the 5' to 3' direction, and it's a very easy process. Okay, so we can just keep going and adding in as helicase breaks apart. We can just keep adding in the 5' to 3' direction using DNA polymerase. Okay. So replication of the leading strand is simple as the template, the parental strand. So here is my template strand, the one at the bottom, okay, or it's also called the parental, is oriented in the proper orientation, 3' to 5', to allow new bases to be added in the 5' to 3' direction. So step one, the first thing we have to do is we have to add an RNA primer. This is only going to have to happen once in the leading strand. So we add an RNA primer. So the RNA primer would be like right here. It's not labeled on here, so I kind of just drew it in here. So we add an RNA primer to the beginning of the leading strand, which happens to be at the origin of replication. It only needs to happen once. Then DNA polymerase 3. So focus on this number. It's 3 because we have DNA polymerase 1, we have DNA polymerase 2. But the one that adds bases is DNA polymerase 3. We'll now start adding nucleotides in the 5' to 3' direction. So then we just keep adding nucleotides all the way through here. Okay. So a very simple process, only 2 steps for the leading strand. Now let's go through the lagging strand. Replication of the lagging strand is a lot more complicated. The problem is the parental strand is in the 5' to 3' direction. So here's my parental strand, and it's in the 5' to 3' direction. Okay. Well, DNA polymerase is going to have to work the opposite direction, so it's going to have to work to the right. So the problem is corrected by creating short little sequences of replicated nucleotides instead of one continuous sequence of nucleotides like the leading strand. So I'm going to make a whole bunch of little fragments here. So I'm going to go through all of these steps, and you can see, there's 6 steps compared to 2 in the leading strand. I'm going to keep going to this picture. So the first part, okay, RNA primer will bond just downstream of the replication origin. So here's my lagging strand here on the bottom. Okay, and the first thing I need to do is attach primers. So that primer, here's a primer, here's a primer. So the first thing that needs to have to come in here is we have to add in this little green spot right here, which is a primer. So we're going to add in a primer. That's the first thing we're going to do. Second part, DNA polymerase 3 will bond to the RNA primer and begin assembling nucleotides in the opposite direction of helicase. So after we've got our primer here, then this is DNA polymerase 3, and it's going to start making little bases. And it's going to keep making bases until it runs into another primer. Okay? On the lagging strand, I can just keep going and never stop. But here, DNA polymerase is going to smash into another primer, and then it's going to stop. So I'm going to have a little fragment of DNA, primer. Then I'm going to have a little fragment of DNA, and then DNA polymerase is going to keep going until right here it's going to stop, and I'm going to have another primer. Okay. Second part, DNA polymerase will stop at the origin of replication or when it runs into an already laid sequence, like we did here. Okay? Or, sorry, when it runs into this primer, it's going to stop. Okay? RNA primer will bond just downstream, closer to the helicase, and the DNA polymerase will add more nucleotides. So as this thing keeps breaking opening, I'm going to have a new primer. A new primer is going to bond like right here, DNA polymerase is going to bond right behind it, and then I'm going to start making bases until I run into this primer. DNA helicase is going to open up a little bit more, then I'm going to attach a new primer. So in the lagging strand, I'm constantly attaching primers, and I'm constantly attaching DNA polymerase 3. Very unlike the lagging strand, it only has to happen once. It has to happen millions of times when we do it on the lagging strand. Okay. So this creates a short little sequences called Okazaki fragments. So if I go back here, these little segments here. So here's a segment, this would be a segment, there'd be a new segment coming up here. All these little tiny pieces of DNA we call Okazaki fragments, okay, named after the Japanese scientist. So RNA primers are then removed. So we're going to eventually go in and we're going to remove all of these primers, and there's going to be, again, a ton of them. There's only 2 shown here, but there'd be one here, one here, one here. There's going to be another one here. So we've got to remove all of these primers. They're going to be coated with it. There's going to be a ton of them. So the RNA primers are removed and replaced with the DNA bases by DNA polymerase 1. So there's polymerase 1 now acting. So polymerase 3 adds the bases. Polymerase 1 removes the primers and then adds DNA bases. Okay, and then the last step that we have to have is Okazaki fragments are joined together by DNA ligase. And the last thing we're going to do is we're going to take this molecule called ligase, and here's an Okazaki fragment, here's an Okazaki fragment. Ligase bonds it together. Okay? So when this one smashes into this one right here, ligase is going to come together and bond them together. Or when this DNA sequence comes in and smashes into this primer will get replaced by actual DNA molecules, and then ligase will come in and seal these pieces together. Ligase is kind of like the glue, where you're going to take all these Okazaki fragments and then glue them all together using ligase. And now this is a really good figure here, and you may want to print out this figure, where it tells you 1, this is the step, then 2, then 3, then 4, then 5, then 6. Okay, so that's the lagging strand. Now the last phase that we're going to go through, because again we have 3 phases. Okay, we've got the initiation, the elongation. Now we're going through this last phase here now, the termination phase. So we've already replicated DNA, and now we've got to get this thing to end. So as soon as the DNA has been replicated, okay, and we got some replicated DNA down here, it winds back into the double helical structure. And the replicated machine, which is the combined efforts of helicase, ligase, the polymerase, and all the other molecules, is disassembled. And we've replicated our DNA. Okay? Now here's another good link that I highly suggest you going through. So it's kind of an animation of going through what I just talked about. And again, I know you're like, well, I don't really want to look up this link. Look it up, okay? It's all these figures I've gone through, and I think they're excellent. Okay? So what I'd like you to do today is I want you to go through correcting errors in DNA replication. So there's probably some content in the content section on how we correct errors in DNA replication, or you can look it up, but it's something I expect you to know. Right? Also, look up some videos and animations on DNA. Go to Google, DNA replication, okay, or DNA replication animation, or go to YouTube, DNA replication. And I'd like you to find some good videos. Don't take the first one. Find some good ones. And then this would be a pretty handy thing to have. I think you should make a chart in your notes, in your portfolio, where you kind of have, well, what does helicase do? What does gyrase do? What does RNA primer do? What does ligase do? What does DNA polymerase 2 do? What does DNA polymerase 1 do? What does DNA polymerase 3 do? So I haven't completed a complete chart here, but it's nice to have this little handy chart so that you know exactly what the function is of each of these. Okay? And then there's kind of our picture, which kind of has everything that we kind of talked about. You should know everything what the job is on all of these things. DNA primase, DNA primer, ligase, polymerase. Is this polymerase 1 or is it 2 or is it 3? Helicase, single standard binding proteins, topoisomerase or gyrase. Okay. And that's the lecture on DNA replication. Okay. Now I have a worksheet for you too. The hardest part is really just that 5' to 3'. So really, spend some time looking that over, and if you're confused, look it up in Google. What is the meaning behind 5' and 3'? It really comes down to the orientations of the sugars, but that's the big focus on DNA replication. Okay, and again, the hardest part is the lagging strand. So some of the questions that I'll probably ask you about DNA replication will be kind of things like, being able to give you a strand and saying, okay, well, tell me how this replicates. I might ask you about, the function of different proteins, like tell me the function of helicase, or tell me, what wouldn't you be able to do in DNA replication if you didn't have DNA polymerase 3? And you'd have to answer, well, without DNA polymerase 3, I'm not going to be able to add any new bases. Okay, so if you have a textbook as well, do some questions out of the textbook. You really should have a biology textbook. Anyone works because we all have the same information, but it's pretty handy and it's more a little comforting to actually have a textbook and then to have those questions that I can get done. So you also got your 2 assignments that are due tomorrow. So make sure you get those in again anytime before midnight. And I hope to get those marked. You also have those 2 optional assignments. So some people have handed them in. I'll try and get some feedback for you this weekend. About if you want to do a DNA diagram to draw one out, and I'll give you feedback on it whether or not you have enough detail. And then the big thing you probably definitely want to do is what you think the contributions of all these scientists are, because what you think the contribution of the scientists might be different than what I think. So if you write me a paragraph or a sentence or 2 about what you think each scientist is, and again that was all in my previous lecture, I might give you some feedback on, oh well, you missed that. Rosalind Franklin did not only the fact that the bases are on the inside of the DNA, but she also talked about it as being symmetrical, and she also talked about measurements. So, wanting to make sure that you have a good idea of what each scientist did in their contributions. Okay, so that's it for today. Hope you guys found this helpful, and we're going to be moving into probably the most exciting thing about DNA next, which is the protein synthesis, and we're going to be able to start decoding DNA. So we're going to be able to take all these letters and now actually take the letters and turn them into protein. And we're going to show you the universal DNA code, which is just, it just blew my mind. I think it's so amazing being able to predict, exactly what protein is going to be made based on our DNA sequence.